So this is essentially the last topic of this course, and that has to do with structure. If you remember, one of the papers that we talked about in the first ranking author was a Mark Gerstein, and what Mark Gerstein indicated in that paper was that bioinformatics really was a confluence of the genes and the proteins, and eventually, when it came times to applicability, for example, in the clinical domain, the structure was important. Okay, and that is what we're going to talk about here. So we're going to look at the DNA structure. We're going to look at RNA structure. We're going to look at the structural nuances. So this is purely from a structural standpoint. And then we're also going to look at aspects of the protein structure. And then we will look at the protein data bank. Okay, so we have here a single strand of DNA, and we looked at the DNA structure at the beginning so that you got an idea of what you needed to do in order to learn about DNA. Okay, so this is a single strand of DNA. You go from the 5' end to the 3' end, and notice something, the lack of, on the 2' ribose sugar, the lack of a oxygen is why it's called deoxyribonucleic acid. This is the ribose sugar backbone, the 5-membered ribose sugar and ribose sugar backbone. And also they're connected by phosphodiester bridges. And it's called ester because the oxygen is a linking atom. Okay, a linking atom. So there's one ester and another ester, and it's these two ester bridges, or the diester bridges, is what links the ribose sugar backbone. We have the nucleic acids, and you can tell that guanine, cytosine, thymine, and adenine. And in terms of these four, and we know about the complementarity of G with C and T with A, and guanine and adenine are purines, so they are a fused ring of a 6-member and a 5-membered ring, so the purines. And then you have cytosine and thymine, which are 6-membered ring, which are pyrimidines, and a purine pairs with a pyrimidine, and that's why A and T and C and G. And this is illustrated in the in the double helix, so where the complementarities are paired, and you will see that the adenine and the thymine, you see the interactions occur via hydrogen bonds, and we've had a discussion about that. So 2 hydrogen bonds, and the interaction between the cytosine and the pyrimidine here, and the purine guanine here is via 3 hydrogen bonds. Okay, and this is what justifies the double helix. Now, for example, you can look at this DNA. You can look at the complementarities always there. Okay, and you can look at these, it kind of splits into single, and then it finds another DNA strand with the complementarity. So that is the DNA structure. If we bore a little deeper into it, you see this is kind of a stylized presentation of the sugar phosphate backbone and the structures. And what you will see here is between base pair and base pair, it's 0.34 nanometers. This is called a minor groove. You can see the groove in the turn. And then the deeper ones are called the major grooves. So between any 2 minor grooves and any 2 major grooves, the traditional, typical, what is called a B helix is about 10 times the distance between base pairs at 3.4 nanometers. And if you represent a ball and stick representation, so you actually get an idea of the atomicity of this is what it looks like. Now, there is not just one type of DNA structure. There are 3 types of DNA structure. And so the traditional one that we see is what's the standard double helix, which is called the B type. The one which seems a little, seems that its structure is distorted is the A type. And so you might have the A type distortion is formed like an RNA DNA or an RNA RNA, getting together would form the A type helix. And then you have the Z type double helix of DNA, which doesn't look that different. There are some structural features that are different. It's different from between the B and Z, except that the Z type is a left-handed coil. So for example, we have a right-handed coil, and you see the way in which the mouse is moving versus a left-handed coil. Okay, some structural features, it's right-handed, left-handed. The number of base pairs per turn is 10. Remember, 0.34 between base pairs and 3.4 nanometers turn. So you will see that there's more base pairs, but it's a vertical rise. So between grooves is 2.56 nanometers, which means that it feels like it's really squished. Okay, and this is 12 base pairs per turn, and that's 3.7, so slightly elongated. And the rotation, remember, this is a double helix. We tend to see it in 2 dimensions, but we have to really look at it in 3 dimensions, and that is the rotation per base pair is 36 and negative 30 because it's left-handed. Okay, and so the handedness tells you what the, but 36, 33 is the rotation per base pairs, these many degrees, and the helical diameter, so 19 and 18, as you can see from the picture, other than the handedness, the Z and the B look the same, but because of its kind of flattened nature, very highly distorted nature, you see the helical diameter is a lot bigger in terms of angstrom units. So DNA is formed in several different ways, but one of them, which stays within the helix, double helix, but looks, but you see a different kind of a DNA structural representation, and that is what is called the Holiday junction. And the Holiday junction is, it is formed during DNA repair, for example. Okay, when the DNA is repairing at the same time the structure. So you think in terms of the complementarity, and this is kind of a schematic type of representation. The complementarity is still maintained, but it forms a junction. So four of them get together, four strands get together. Now, in real life, these are actually double helices, but these helices kind of, they break apart at this point, and this is mostly for genetic recombination DNA. And remember, the base pairing is still preserved. When you have something like this, it means it's a twofold axis of rotation, which means think of it as looking directly into the plane of the screen. Okay, and then you look, which means you're looking in this direction, and the Holiday junction does appear as if kind of two double helices are getting together to form some sort of a superhelical coil. But if you then look at it differently, like if you look at it from this view, you'll see that there is a Holiday junction. Okay, so notice something, that you have the 5' end of blue, and it becomes the 3' end of blue, which then forms the 5' end of green, and you can see the color coding tells you how the helices are formed. The complementarity, as I said before, is still maintained, and this is a proper kind of a stick representation, which tells you what the this is what the atomic level is. Now, what you see here is continuous strand. These are the continuous strands, for example, 5' to 3', 5' to 3', and that's how it goes. And so you see the Holiday junction. So this is what is the, a little bit of that unpairing occurs when creating the crossing strand. This is what this side is called the continuous strands. Now, it feels like, despite the fact that it seems that they're 90 degrees to each other in the schematic, they really are not. So this is at 101 degrees. So this is a little more spread out, and this is a little more put together. And then you have what are called sessile phosphates. Sessile phosphates are covalent bonds. Okay, that's the definition. Covalent bond that is subject to be broken by an enzyme. So susceptible, not subject, but susceptible to be broken by enzymes. So these are called sessile phosphates. Now let's go to the RNA structure. RNA and DNA is basically the same. One would think that, well, other than the fact that there is a missing oxygen on the 2' part of the ribose or the 5-membered ribose sugar, that should be the difference, right? But there's significant structural differences. And there's some additional differences, for example, so the phosphate diester backbone stays the same. There is what is called a pyrophosphate region. And so this is where different aspects of either the nucleic acid or the backbone is modified. Okay, is modified in RNA, and that'll tell you, once you look at the structure, it's really always a single strand, which sometimes doubles on itself in these really interesting ways. The other important thing is that so that's the pyrophosphate region. Then the other part that's different is the uracil, which is different from thymine. So in this case, the pairing will occur between adenine and uracil, and cytosine and guanine. Now, RNA does not form a double strand, unlike DNA. Okay, so it forms this weird kind of structure, but you will find pairing. The complementarity is still maintained. You will find the pairing exactly as it would in DNA. There's some variations, and we'll look at that too. Okay, so that's how the pair. There's no double helix. It's just single helix, and there is a reason why. Now the specific reason, there's not a lot of reason. The specific reason why RNA formed this kind of a structure is versus why DNA is so stable. This is a good question. Now remember that, and kind of at a kind of a basic biology level, we always think in terms of a DNA. Carries the code of life. It carries the code of life. It is present in every cell in for a particular species that contains the entire code for that life. Right, all the chromosomes, all the genes, everything that constitutes the genome is present in every cell, in the nucleus of every cell. Now, that's DNA, right? On the other hand, RNA is very often it's created in an impromptu way. RNA is created in an impromptu way because RNA basically is created just to affect a particular function. Once that function is affected, then the RNA can go away, and therefore RNA is inherently unstable. Okay, because it doesn't really it's just responsible for a momentary affecting of a particular function. Okay, here's another way of looking at it, and you will see, for example, so this is another schematic way of representing it. There's other things that's interesting. For example, you'll see that some of these bases, they do not look like your typical ACUG um, basis. And so you're going to find something called as a pseudouridine, a dihydrouridine, an inosine, a ribothymidine, a methylguanosine, a dimethylguanosine, and a methylinosine. Okay, and what you'll see here is it's possible that one might, again, there's nothing definitive about why these unusual bases are formed, but you can see just from this representation, they are formed at these junctions. It's almost as if that they are in place to prevent base pairing. And once you prevent base pairing, because the natural complementarity causes DNA to kind of form that double helix. But if you have these breaks, because of these modifications, you will find that these modifications do not allow the base pairing and therefore maintains the structure in a different way. There's something else I want to point out to you here, and that is, notice that G and U are paired, which is not supposed to happen, right? In DNA, it doesn't happen. But here, these are what are called as wobble pairs, that once in a while, that complementarity of AU and CG, there's a violation, and these are what are called as wobble pairs. And this L-shaped kind of pseudo L-shaped structure is typically what the structure eventually looks like. Wherever necessary, there's pairing. Okay, and then there's no pairing, and some of the other the base pairs, or the bases, the nucleotides, the unusual nucleotides kind of break that pattern, causing this particular structure to form. This is what's called the anticodon region. So during DNA protein amino acid synthesis, the anticodon region binds to the codon region, because remember, it's supposed to transport to the site of protein synthesis. In terms of, now this is not exactly 90 degrees, these two are, but it seems that it's slightly skewed. Okay, so this is the RNA structure, and so you can look, for example, at RNA structure from this resource, and you'll actually see that there is complementarity, there is pairing, but that pairing is not narrowly maintained, and it's kind of, there's violations to that because of unusual nucleotides. Okay, and this will actually show you that L-shaped structure. Okay, now let's get to protein structure. So in proteins, for example, let's look at what proteins constituted of, and we have the 20 typical amino acids, and it's not easy to remember some of these amino acids in terms of their structure, etc., but you do have an overall view, as to some basic aspects of it. So, for example, these are the three-letter code, and then each of them is represented by a single letter. But here's one way you can remember these. There's what's called the non-polar groups, the polar groups, the non-polar amino acids, the polar amino acids, and then the electrically charged amino acids, which are negative and hydrophilic, and then we have the electrically charged, which are positive and hydrophilic. Okay, so lysine, arginine, and histidine are the positives, and aspartic acid and glutamic acid are the electrically charged but negative, and serine, threonine, cysteine, tyrosine, asparagine, and glutamine are, and this probably because of the presence of a heteroatom, nitrogen, oxygen, etc., which allows it some sort of polarity. And then glycine, which just has a side chain of an H, etc., are these are non-polar. Okay, this is a great resource. It's a nice little handy thing if you want to know something about the structure. If you want to know something, and I won't go into that right now, but if you want to know something about the structure, if you want to know something about certain properties of amino acids, this is a great resource to have. Okay, so in terms of the structure of amino acid, again, it's not you can't really remember all of this, but you think in terms of the arginine, histidine, and so you see that a lot of the nitro groups, not the nitro group, the amino groups, whether it's a secondary or a primary amino group, is what lends its the basicity. Okay, and in terms of the acidic group, which are aspartic acid and glutamic acid, the dicarboxylic acidic groups is what renders the acidity. So it's the amino groups and the carboxylic acid groups. And so if we have, for example, we have glycine, which is just an H group. Now this is the amino acid, amino and the acid group for the amino acid, and this is the hydrogen. And you can look at what the structures of each of these are. And remember, for example, if you have this thick, slightly triangular, it means it's coming out of the plane of the board. And if you have a dotted line, it's going into the plane of the board. This is from the stereochemistry. So you kind of, it's a two-dimensional representation, but you understand the three-dimensionality of these amino acids. Okay, so in terms of the basic formation of the amino acid, you have an amino acid with the amino group, and the carboxylic acid group, and you lose a water from the OH from the carboxylic acid and one of the amino groups, the hydrogen from the amino group. You lose a water and you form a peptide change. This is the amide bond, CONH, or also called as the peptide bond. And several of these peptides will get together to form polypeptides. So it's essentially a polymer composed of one or more types of amino acids. So again, the CONH group is a peptide group, is the peptide bond or the amide bond, and the R groups are the only things that are different about amino acids. Okay, and so this is the formation of a peptide bond. In terms of the protein structure, when we refer to the primary protein structure, we refer to a sequence. So it's amino acid sequence. And there are features which cause these amino acids to form into secondary protein structure. So you have pleated sheets or beta sheets, an alpha helix, right-handed alpha helix, and the third part where there is no defined structure, for example, is what is called a loop. In a tertiary protein structure, you'll find that all the secondary structure elements combine together in a fold, and that is called the tertiary structure. And the protein folding is one of the biggest challenges in science at this point in time. And then you have what is called the quaternary protein structure, where several subunits, which are kind of individual proteins, will get together mostly for functional reasons. Okay, so the primary structure and you can write this in the three-letter code as well as the one-letter code. The secondary structure is stabilized by hydrogen bonds. It's a hydrogen bond. It's not a real bond in terms of the exchange of electrons, though it's a bond caused by by the borrowing of electron density. But it's really the foundational bond for life, right? I mean, you wouldn't have double helix in DNA to preserve the genetic code of a species would not be available without a hydrogen bond. Similarly, protein structure, the three-dimensional structure for protein, is not possible without a hydrogen bond. And so where does a hydrogen bond take place? The hydrogen bond is essentially between the so think about it as CONH, okay, CONH. So this is nitrogen, NH. So between the H of the nitrogen and the oxygen of the carboxylic oxygen of the amino acid, and that's where the hydrogen bond takes place. And the strength of the hydrogen bond is determines whether something is the alpha helix or A beta sheet. And when you have these hydrogen bonds, for example, these hydrogen bonds are in a very strategic way between the CO double bond and the NH hydrogen is depends upon the strength of this. You can actually have um, proteins, amino acids forming particular secondary structure. And so this is what it really looks like. Okay, so you have R1, R2, R3, and you actually have a coil, like a kind of a spring coil kind of a structure. So this is what the secondary structure looks like for an alpha helix. There's something you have to also notice is what is called a helical wheel. So a helical wheel is, if you look at a helix, from its geometry and its structure, let's say, for example, this is a sequence. If you look at a helix from the top and you just kind of look at it right like top down, you'll find that it feels like it's a wheel. It's a circle. Okay, so the question then is based on how the turns of the helix, the coil of the helix, and the spring looking aspects of the helix, you'll realize that how is this presented? So imagine you have a circle, you have 360 degrees, and they say that when you look from top down, you look at the first, let's say, and you place the first amino acid at position 0 or 360. The next position, which is look is tryptophan. Tryptophan is 100 degrees away. Okay, and if you look down it, if you represent the three-dimensional wheel in a single dimension or two dimensions or on a piece of paper, that is where the second position. So it's almost like they are positioned at angles that are 100 degrees away. So this will be 0, this will be at 100, 3 will be at 200, and 4 will be at 300 degrees. Okay, on a 360 degree wheel. And then 5 will be, because this is 300, this is 300 and okay, this is 300, this is 400, and so 400 is F. Okay, and then there'll be 500. So that's what it looks like. So it's a two-dimensional representation. And what happens when you're done, when you're done with 1 2 3 4 5 6 7 8 9 20, when you're done with 20, you just write it on top. So that's why this K is here and this F is here. Okay, and that's a representation. Now, because notice something interesting though, because a helix has a hydrophobic side and a hydrophilic side, right? It's amphiphilic or amphipathic. You will find that even when this position, when you look at this in the helical wheel, you'll see that for the most part, one side consists of hydrophilic charged residues, and the other side consists of hydrophobic or non-charged residues. Okay, now just like there were different types of DNA, there are different types of helices. Okay, and the basic helix that we're talking about is what is called the alpha helix. That is what we see. There's another one which is called the 310 helix, and a third type which is called a pi helix. So, and these are the pictures of that. How many residues per turn? 3.6, 4.4. So the pi helix looks like it's stretched. The 310 helix looks like it's kind of compressed. So the translation per residue is 1.5 angstrom units per residues, 2 and 1.1. And the radius of the helix, this is again, while it seems compressed, it's also compressed in along the kind of the vertical axis. Okay, and so this is 1.8, whereas the pi helix is stretched, but though it's stretched, it's also pretty wide. Okay, so it's 2.8. And these are the pictures of the pi helix, the right-handed helix, which is traditionally what we look for, and then the 310 helix. Now, in terms of beta sheets, you'll realize that the hydrogen bonds are the same, except the hydrogen bonds tend to be weaker, and the you can see the directionality. So these are anti-parallel beta sheets. So this is the amino acid. This is the amino acids. Kind of, if you trace the secondary structure, those are the amino acids. And but the position is such that you have these strategic hydrogen bonds, and these hydrogen bonds, because the nature of the hydrogen bond, they're not as strong, so they don't allow it. They feel like it's kind of a stretched helix. It's a stretched helix, and because it looks like that, they're called beta sheets or pleated sheets, like this, like very regular folds in that particular sheet. Okay. In terms of purely structure, we are looking for something called as a torsion angle. And what's a torsion angle? Now, typically, an angle is decided by 3 points, but a torsion angle is typically decided by 4 points. And so, what is the 4 points? So, let's say the way you do it is this. That's why. So, for example, let's say you have a plane, which is which is defined by A, B, and C, and another plane that's defined by BC and D, where the segment BC is common to both. So the angle between the two planes, okay, the angle between the two planes defined by two different angles is called the torsional angle. So if you take A, B, and C as a plane, A, B, C as a plane, and B, C, D as a plane, the angle between the plane, and you can try to do this. For example, you can take a folded, piece of paper, fold it along the middle, put A on one side and D on the other side of the fold, and put B and C along the fold. And then you see that the angle of that fold is then the torsion angle. All this brings us to something called as the Ramachandran plot, something that had been determined a long time ago. And that is, when you determine the structure of a protein, you look at that protein and you study the torsion angles, and then you plot the torsion angles of that with each other. And if they are supposed to be um, kind of it's if the protein has no structural violations, for example, like things that are atoms that are pushed together that do not belong should not be pushed together, etc., it is what is called as a Ramachandran plot. So look at all these places, right? So, for example, what you see here is what's called the pi torsion angle. And the pi torsion angle is formed between, for example, the planes, um, the NH group, okay, the CO group, the CONH group, the peptide bond group, and this plane between the C alpha. That's the first carbon atom of the side chain. Okay, that's the side. So that angle between these two planes, see this plane here, this plane, and this plane is called the pi angle. So this torsion angle can be calculated. And then you calculate the next one, which is called this is psi. I'm sorry, this is psi, and this is what's called phi. Okay, this is psi, and this is called phi or phi. And you see that this plane here between C alpha and the nitrogen, and then this plane, okay, that angle between these two planes is called the phi or the phi angle. And then there is another one which is called the omega angle. But you plot the phi, the psi and the phi's versus each other, and you'll find that where do most of the angles lie. So, for example, if it's a regular beta strand without any violations or conflicts, then it should lie here, and that's the maximum. And then the alpha helix should lie here, and this is, I think, the 310 helix. And you'll find, very few potential outliers. And if, for example, if your angle falls in these white areas, which means there's something not right. It means that the structure solution of your protein is incorrect for in that particular respect, because that means those two planes which define the angles are not are there's a violation in those planes, which means the structures are the atom assignments are not correct, because that pushes atoms too close together. Okay, and that becomes energetically non-favorable. You can also think of it in terms of what's called steric repulsion. Steric repulsion is when atoms are too close, then they kind of these two atoms tend to repel each other, and that's why stable structures are structures with very little steric repulsion. And so, for example, if you think in terms of what kind of structure does a compound adopt, it adopts a compound in such a way that the heavy atoms are always on the other side, like trans, as opposed to on the same side because of the steric violations. Okay, again, what is steric hindrance? Some values of phi and psi are forbidden, since some atoms will come too close to each other, resulting in what is called a steric clash. For high quality experimental structure, these regions are usually empty or almost empty. That is these regions. Okay, so if you find phi and phi and psi angles in these regions where not so, the best is where they are in these in the highly populated regions without there's no problems. But if they're found in these regions, then there's a problem. Very few amino acids have their torsion angles within these regions. Now, there have been stable compounds, properly determined structures of proteins that violate the Ramachandran. So the kind of the knowledge that went into Ramachandran, into the creation of the Ramachandran plot, for example, is probably a little out of date. It's mostly good. You can identify the ones, the potential violations, but some families of proteins, for whatever functional reasons, do adopt these structures, do adopt some of these torsional angles which violate the Ramachandran constraints, but they are still functional and they are still stable. There are exclusions from this rule. Sometimes such values can be found and they most probably result in some strain. In such case, additional interactions will be present to stabilize such structures. Okay, so that's something to take in mind. And these might have functional significance, and some of these might be conserved within a protein family. So a particular protein family affects a certain function, and therefore these proteins, so to speak, can live with that additional torsional strains. Okay, what about glycine? Now, glycine, in terms of the phi and the psi angle, you find it all over the place because there's no torsional strains because it just has a hydrogen atom, right? Remember, most of the phi and C angles involve the C alpha. The C alpha is the carbon atom attached to the C double bond O, which is the beginning of the side chain, which defines the amino acid. Okay, so that's glycine. So this is the typical Ramachandran plot, like a beta and alpha, and then maybe the 310 helix region. But you'll find glycines everywhere, and they're not really considered violations because there's greater freedom for the torsional angles for glycine. In terms of the tertiary structure, you'll see the red are the helices, the beta sheets are in green, and the loops, different kinds of loops, for example. Where multiple subunits of a protein are arranged in 3 dimensions with respect to each other is where we get a quaternary structure, right? So primary, beta sheet, alpha, this is the tertiary structure, and different subunits get together to form a quaternary structure, and is mostly for functional reasons. For example, hemoglobin has what is called a heme group, which is an organometallic region, an organic group, all what's called coordinately bonded to an ion. Okay, and so that really this is really an alpha chain subunit. Okay, there's 2 alpha chains, there are 2 beta chains. Okay, and so each of these are a separate subunit which is own tertiary structure, but all these get together into what is called a quaternary structure. And you remember this ion then binds with oxygen, and it is the oxygen transport in blood, much like oxygen transport in myoglobin is but myoglobin has only one subunit. It doesn't have a quaternary structure, but it also interacts. So myoglobin will be the oxygen transporter in muscle. Okay, and this is the quaternary structure. This is the heme group. Okay, this is the heme group coordinated with iron, and you see there are 1, 2, 3, and 4 structures. There's something else that you should know in terms of structures, and that is what is called the disulfide bridge. So disulfide bridges are formed only with cysteine, where there is a replaceable hydrogen atom here. And so you must remember that even, for example, if there are cysteines in 2 remote parts of the proteins, they will get together. To form this disulfide bridge. There's hydrogen released, and the disulfide bridge, and this is really important because because that is important for the structure. So that there's consequences for structure formation there. Why is cysteine so important? As you can see, there's if you remember our PAM or Blossom matrices, uh, cysteines are conserved, and you look at these numbers, right? There's fewer cysteines, but they're largely conserved. So a cysteine score is really high. Cysteine score is really high, probably trumped only by tryptophan. So the very high conservation, that means the chances of them being replaced are very little, and probably the disulfide bridges help stabilize the protein structure, which again is important for function. Okay, so this is the PAM 250 matrix. Okay, and that's the cysteine. Why does methionine not form sulfur bridges? Because there's no replaceable hydrogen on the sulfur. This is all already associated with a methyl group. In addition to secondary structure, we have what are called loops. Now, traditionally, and for the most part, loops do not have a definable structure stabilized by hydrogen bonds like alpha helices or beta strands. But depending upon the loops are attached to, what the loops are attached to, for example, they could be a beta turn, beta sheet turn, which attach to beta sheets together, or to alpha helices or an alpha helix to a beta turn. But because these are, very traditional, these are not, then, but it's not just coil anyway. The proximity of these helices or the helices in a strand or two beta sheets, for example, the proximity allows the loops to actually have some structure. Okay, so we have what is called an alpha turn. The end residues are separated by 4 peptide bonds, and because of their proximity, hydrogen bonding is possible. A beta turn is I, and it interacts, is I and I plus or minus 3. Okay, that's a beta turn. We have a gamma turn here. Sterically unlikely, because to have a tight turn will cause all kinds of steric issues. There's a pi turn where they're separated by 5 bonds. And there is a hairpin is a special case of a turn in which the direction of the protein backbone reverses and attaches to secondary structure elements. Now let's look at the last part, and that is the protein data bank. Okay. Uh, when you look at the protein data bank, it is about the solution of a structure. Now you can teach a whole course on X-ray structure solution. You could potentially teach 2 courses, okay? But if you go to this link, you will get a very good idea about the summary of the entire process. You have some idea of the secondary structure of the protein, and essentially what comes out of an X-ray crystallography instrument after analysis is a picture of the electron density. Okay, a picture of the electron density, and it's kind of a smeared electron density. And what you have to do is position your structure, position your primary structure based on these electron densities. Now, there is something called as resolution. Okay, so resolution is the when it says the distance of 5 angstrom units, it means that this is resolved in such a way, this structure is resolved in such a way that you really cannot, anything below 5 angstrom units, you cannot say with any kind of certainty. And you notice something, just to make a little better sense, is you look at the worst where you're trying to put your atoms where in this kind of smear density. But if it was highly resolved, here you can only, you cannot tell the differences in less than 5.5 angstrom. But notice how the densities are so perfectly resolved, then you can actually put your positions of the atoms here. Okay, typically you see somewhere between 3.5 and 1.4, and the better the resolution is, you can place your atoms with greater precision. Okay, and so what you get out of an X-ray machine is just this net surface kind of thing, and in the structure solution process, you actually place your atoms in where they belong, where you think they belong, because you already know what the primary structure is. And again, the better something is resolved, the better it is. Let's go back to the protein data bank. Now, when you look at the protein data bank, this is the first thing you're going to see. You get all kinds of information about, the paper with, the picture of the things like that. But one of the things that this is relatively recent, probably the last 3 or 4 years, is they are giving you a kind of a picture of the quality of the structure. Okay, and we'll look at what these do. So what you'll see here is the more some of these are towards the blue side, that means it is good. The more some of these numbers are on the red side, which means it's worse. So this is percentile. Then where does it stand? How does it compare to all X-ray structures? So in this case, you would say that most X-ray structures are better than this on based on these criteria, these parameters, these metrics, right? And percentile relative to X-ray structures of similar resolution. Okay, so that is important. So this is a poor structure. Notice that everything, this is one thing, percentile of this outlier, and we'll see what that is directly in the blue zone. Everything else is poor in the red zone. On the other hand, something that is a good structure, all these values are going to be in the blue zone and preferably on this side, right? So notice here, for example, we look at the other parameters, but look at Ramachandran outliers. So how many of these atoms, how many of these amino acids violate Ramachandran? In this case, it's zero. And in the previous case, you would say it's really bad because, 14.5, that's a lot. That's like 15% of these are violating, which means they are in the wrong, their position, the structure is not properly solved, and you have all these violations. So you look at these parameters: R-free, clash score, Ramachandran outliers, side chain outliers, and RSRZ outliers. So what is an R-free? This is measure of the fit of the model to a small subset of experimental data which was not used in model refinement. Now, what do you mean by model? In terms of experimental data, this surface is what you see. Your model then fits the experimental data. Okay, and how well it fits is the R value. Okay, and the experimental data was not used in the model refinement. What is a clash score? Is derived from the number of pairs of atoms that are unusually. That's the steric repulsion that we talked about, the steric condition. So what is the clash score? What are the Ramachandran outliers? We understand that. Side chain outliers. Protein side chains mostly adopt certain combinations of preferred torsional angles called rotamers or rotameric conformers, much like their backbone torsion angles. So this is about the backbone. Remember, therefore, the phi and psi angles have to do with the alpha carbon atom, the carboxylic oxygen, the amino acid, the amino group of the peptide bond. But that's the backbone. The side chain outliers say that you have to have certain preferred positions. For example, a histidine is supposed to have this particular position, and they're called rotamers or rotameric conformations. If they and there is only certain preferred torsion angles. So if they violate these, then they're called side chain outliers. And RSRZ outliers, the real space R value, is a measure of the quality of fit. Once again, how well you can put your atoms based on the density that you see. So this is a structure of the bovine rhodopsin, and now we begin to understand 2.2 angstrom units, which is actually not bad. It's a signaling protein. It's a membrane protein. So this is a schematic representation of this protein. These are the clash scores, the Ramachandran outliers, etc. So that's that information. This is the work that was done in 2004, and all kinds of other information related to what families it belongs to, what is the secondary structure. This is the SCOP is what's called the structural classification of proteins, what type of protein, what family it belongs to. Validation, there is an A, there's obviously, this is a dimer, it crystallizes as a dimer. This tells you where it is in Swiss prot, in XPASY. It tells you the molecule is rhodopsin. It has two chains, each of length 349. Bos Taurus. Bos Taurus means it's cow. And you have a gene view. There's all kinds of very interesting information. These are the ligands. And sometimes when you have membrane proteins are very poorly behaved. And so when membrane proteins are very poorly behaved, they often co-crystallize them with other compounds in order to increase the solubility. So a lot of these compounds are charged compounds. And you have, again, structural validation, etc. Again, this is what is called the unit cell. This comes from basic crystallography. What kind of space group resolution, we know that. This is the free R value and the R value work, which is the fit of the model to the density. And we looked a little bit at that. Again, I don't want you to, you don't have to be an expert on this. Certainly, we're just doing a superficial view of some of these things. But if you've not looked at the PDB, this is something that's of interest. Okay. Now, you can download files in different formats. The PDB format, you can download, for example, the FASTA sequence, but you can similarly see everything you need. So, for example, this is a FASTA sequence of both the chains. One of the things that I would like you to see is what's called the PDB format. Okay. Now, the PDB format it'll tell you a lot of information. Most of this is not very useful. What do I mean by that? It's useful to know something about the crystal structure, but, and again, you can teach a whole course in crystallography on this. But, uh, so I'm not going to be terribly interested in this except for one little part, and that is this tells you about the sequence, it tells you about the crystallography condition, it tells you about the things like point group and space group and what kind of heteroatoms, etc. are there. The part that I'm interested in, it also tells you, for example, what residue to what residue is a helix, what residue to what residue is a beta sheet, etc. Okay, a lot of this information. Now, typically, if you look at this and if you put it into a protein visualization program, it's going to ignore all of this. But what I want you to focus on, if there's a heteroatom, that's called het atom. But what I want you to focus in terms of the PDB is this. Okay. So for example, this tells you this is atom number 4. It's N. N just means it's your amide peptide bond nitrogen, the one connected to NH. It tells you what the residue is. Okay, the residue is methionine, start codon almost. It tells you it belongs to chain A. This tells you the amino acid number, so this is residue 1. This, now remember, this is structure, so we're looking at the position of the atom, right? So it's position 1, X position, X, Y, Z, that's where the atom is. This tells you that it is, there is only one of this. Okay, and that, now you might say, well, what does that mean? Shouldn't just there be one atom? Remember, when we create the model of some of this, we are assigning atom to the smear density, like those spheres, those net spheres that we saw, right? But sometimes what happens is we know that that particular atom is there, but the smear is so wide that we don't know where to place that atom point. So sometimes you might find a situation where you say, well, this particular density related to this atom is so smeared, I'm going to put 0.5% here and 50% here and 50% here. So, for example, one atom can have two places, but obviously you can't count it as two atoms, so you're going to have 0.5 and 0.5. Now, what's 47.1? These are what are called as Debye-Waller parameters, and you can look that up, or called as temperature factors. Now, Debye-Waller, remember something, that every structure, even though it's a kind of a solid crystal, for example, has, even at absolute value of 0, has a 0 point energy. So there's some vibrational motion associated with it. And certainly you in when you look at a protein, it's a living viable entity. And this living viable entity is also in a solvent, right? So there is a lot of motion associated with that particular atom. And so this 47.11 is a representation of the motion. Okay, and more the density is smeared, your structure is going to be less good. Okay, and so, and remember something, that we always liquid helium, we always when the crystal structure is being solved, you're always having a continuous stream of liquid helium or liquid nitrogen being poured onto the crystal to keep it viable, and also to prevent the unusually large motions. And that's all I have to say again in terms of of the PDB, is that this is what you really have to remember. Okay, let's look at one slide associated with it. This is the atom. Okay, that's the atom number. That's nitrogen, which is in this case it's the amino the of the peptide group. Tells you that's a valine. Tells you that's chain A. Tells you that is the residue number, which is slightly shifted. It's actually here, position XYZ. Okay, XYZ position. This is what's called the occupancy. That in that particular density, what kind of atom, you want to assign that point to the whole atom, or if it's so smeared, you want to say half an atom here, half an atom there. That is what is called the temperature factor, and that is what is called the generic atom type. It's not very useful because you know exactly what that is. Okay, and I'm going to stop here as far as it's a quick representation of structure. We've been through DNA, RNA, different aspects of structure that you may have not known, but the important part here is looking at the PDB.